Okay, very good. So Dave Thirumalai is our speaker today. Dave is new to this department. He never visited us before, apparently, but he is not new to Manhattan because he was postdoc, right? Not graduate student, but postdoc in Columbia University, which is, of course, a very far neighborhood of Manhattan, but still in Manhattan. Dave's most famous works and most cited works are about glasses, but after that he worked on a variety of biophysics-related topics, too many to list. Recently, he worked on chromosomes, and Alexandra and I were citing his papers quite extensively. So there are many other things that I can tell you about him. He received recently a Langmuir Prize of American Chemical Society for Physical Chemistry.
APS.
It's APS? Yeah. Sorry, I thought it's...
It's okay, it's the same thing.
Yes, AS anyway. So without further taking this time, let's listen better to what Dave will tell us. Please.
Thank you very much. I want to thank Shura for inviting me. I've known a lot of people who are in the soft matter group. I think that there's only two people I hadn't met before. One is David Greer, the other one is Dave Pine, and Greer I met today, Pine I've not met. So there is a little NYU connection, even though I never visited NYU when I was a postdoc with Bruce. I came to him and he asked me, what do you want to work on? I've been thinking about some problems involving liquid vapor interface. So I said, Bruce, I mean, I know that you've written papers with Mel Kalos, who used to be in the ground here. And I said, I want to run something, maybe you can give me some pointers on what to do. So Bruce says to me, you know, you can work on that problem if you wish. I'm not the kind of guy who will stop you, but that'll be like crossing T's and dotting I's. You won't get a job. So he said, don't work on it. I mean, that was the implication, although you know it said don't work on it. So subsequently he told me something else, I did something else. And therefore, had I worked on that, I might have come to NYU, but I didn't do that. And in the 3 years I spent with Bruce, I never came here except to eat in some restaurants. Whenever I could afford it.
That sounds like a very close connection to NYU.
Let's connect to NYU. So I want to tell you about some things today which are not super new to us, but we're not really super experts in it either. So let's watch this movie. I'm going to explain this movie to you. It's a video image of a gas relation process of zebrafish. And gas relation is something that happens universally in all of us. It's a two-dimensional slice. It's where an epithelial cell, which is a two-dimensional cell, if you wish, undergoes this gas relation process in which it folds itself up, and then afterwards it differentiates into various things like lung cells, or heart cells, skin cells, and so on and so forth. Now, this picture is, to me, remarkable. It's something that we've been only thinking about for the past 3 or 4 months. And you see that there seems to be some kind of a flow, and indeed there is. This is a living material, living species, I would say. It has nutrients it's consuming, it's dividing and growing. But if you look at this movie long enough, or even one time, you'll see that there's some sort of an interface that's over here, and in the right side of that interface, this is a two-dimensional projection the cells are narrowing in one dimension, let's say x, it's growing in the other direction. And this is called convergent. It means it converges in the x direction. Extension means it goes in the other direction. Of course, this is happening in 3 dimensions. But this is just a 2-dimensional slice because apparently experimentalists friends tell me that the 3D imaging is really very hard. Okay? So whatever you think of this movie, it is very clear something super collective is going on, and it's a consequence of cell division and so on and so forth, and other complicating factors. The cell dimension is about 10 or 20 micrometers, and if you simply analyze the mean square displacement of the cell, it's not average too much, so it goes as a power law with an exponent that's greater than 1. In fact, it's super diffusive. So one would like to understand things of this kind. And we haven't quite sorted this out, although we're working on it. Okay, so what I'm going to tell you about is how we generated this movie on the right-hand side, or this picture on the right-hand side. And it's not unrelated to this movie that I just showed you. Its dynamics has some commonalities with the previous picture on the zebrafish development at the gas relation stage. So I must credit all these guys who worked with me. This guy is still here. He's now a professor in Georgia, and this student is a postdoc in Harvard right now. So we means that these 3 guys created this image on the right-hand side. The first question is it a reasonable view of the physical tumor growth? And I haven't told you what was used to create this. And what are the consequences if it's a reasonable model? And that's what I want to explain to you. The diameter of this solid tumor, as it were, is 0.2 millimeters, about 10 times or more smaller than actual tumor that you will see an image of later on in the talk. And this aspects of heterogeneity and the velocities and the positions and things like that will naturally emerge from the kind of simple model that I'm going to tell you about. So what is interesting about living systems, we all know this very well, it spans a length scale of about 10 orders of magnitude, and we're going to be living in this 10 to 20 microns. And at each level of this description, new things had to be included and probably old things had to be excluded just because of the complexity of the problem would increase enormously otherwise. And this goes back to some of the things that the late Phil Anderson in fact talked about, and then Stein and I reminisce about that a little bit. So the trick is, for a long time, I spent working on this nanometer scales, say, or 100 nanometer scales. These are things like ribosome, polymerases, how they function, etc., and these tissues, when you want to work on those, you can't possibly think about molecular scales. You get to something different. So I got into this problem very accidentally when Ted Kirkpatrick, my colleague at Maryland, and I wrote an article in the Reviews of Modern Physics about 8 years ago, where we were trying to initially describe a theory for glasses that we had developed now about 30-some years ago. And we decided that we want to show that some of those ideas may cross over to problems in biology, from molecules to tumor level, and also to problems in condensed matter, including some quantum phase transitions. So it contains a mix of both. And at the time, we knew about things like heterogeneity. In fact, one of the problems we worked on first in 1989, shortly after. Paul probably doesn't remember any of this stuff. And Paul had come to University of Maryland to give a talk, and I think he talked about his Wigner glasses, mixtures of binary mixtures of charged particles in 84. Paul, you forgot? So, I remember. So I was taken in with that paper then, And our first paper on that subject was in '89. We'll be writing one this year. In between, we've written 2 or 3 papers on the same system that Paul and Mike described. And what you find in the glass formation system is now the glass community kind of accepts this, that if you look at, and here of course temperature is not a relevant variable, it's a density or the volume fraction occupied by this big charged particles. That's a relevant variable, and if you compress it long, it undergoes and shows signatures of glasses. I can't quite recall. I know, for example, in the 1984 JCP paper, there were structure factors. I can't remember if there were any dynamics in there. I don't remember that very well. Maybe you do. What you find in these glass forming systems is you start with a homogeneous system in a liquid, and you compress it in this case, and then you find some particles move more than others, and some particles move less than others as relative to the diameter. And this is called the dynamic heterogeneity. And we more or less understand what the origin of that in the context of glasses. This picture here is a 2-dimensional Lorentz fluid simulation. It was reported in this article by in the Reviews of Modern Physics again, 2011, by Giulia Biroli and Ludovic Berthier. And when you see this guy here, you see patterns like swirls and vortices and things of this kind. And here, of course, this is created when you undercool the liquid a lot below its fluid glass transition temperature. So
Can I ask a question?
Yeah.
What violates law of large number means?
So if you take a liquid, then let's say I take a homogeneous liquid, and I ask what's energy per particle. So I take a large enough sample, it contains 1,000 particles, something like that, in some region, and I compute that, measure that, I'll get a number. And I'll get exactly the same number if I went to a different region and took 10,000 particles or whatever. So it's large numbers are satisfied. And that's why computer simulations sort of work. In liquids, you don't need to simulate an infinite system, you need to simulate a discrete system. If you do the same computer experiment in a glass, and we did that again 30 years ago. This is why
this glass, specific glass, this charged.
No, that one we did with 3-dimensional Lennard-Jones fluids, and there it turns out different regions give rise to different distributions of energies, even though the interactions are exactly the same. So a lot of large numbers violated.
Mathematically, to violate the law of large numbers, you need a diverging length. Like as you scale the size of the little window you're looking at, even to infinity, then only that average.
That is true, but I don't have a luxury of simulating infinitely large systems, so we simulated something like 500 to 2,000 particles.
Going all the way to system size.
Yeah. And then we look for 1/n corrections, and did our due diligence to show that it's not mathematically rigorous as we found out. Okay, so these swirls and all that you see here. Now, while we were writing this particle, we came across this picture. I don't know why I was looking at this annual reviews of pathology, mechanism of disease, and I saw this picture. Supposing I cut this bottom portion out completely, I could have told you that different colored particles are different velocities of some system, and this is exactly like in the previous slide. You have classes of particles. But this is an extremely, the previous slide of course totally is irrelevant, who cares about that? But this is relevant. And normally when an oncologist suspects that you have some kind of cancer, he or she will say, let's do a biopsy. And until very recently, I don't know the practice even now, but they would take a small piece of your biopsy, because I'm probably human beings again. Keep chopping the solid tumor everywhere, and then they would find that there's something wrong, and then there's a protocol, there's a device, whatever that is, and that sort of works for a while, but subsequently, the phenotypic or genetic variations that were treated based upon that small biopsy, the thing that's far away doesn't care about that. It's got different genetic mutations, and then the cancer recurs. And once it occurs the second time, it's usually deadly because these cancer cells, which are much more mobile than normal sticky cells, move very far away, and then the disease progression takes place. That's a kind of heterogeneity. In the case of tumors, this heterogeneity could be both due to genetics and also stochastic variations, and both of them had to be taken into account. So when we wrote this article, I knew 0% about cancers, and so that was, let's say, 9 years ago. Now I know 0.01%, and because it's massively complicated stuff.
Sorry, what do you mean by genetic sources of heterogeneity? You mean different mutations?
Yeah, mutations. You know, I'll show you a picture of that in just a second. That's exactly what I mean. So this is a remarkable article by this person, Gloria Hepner. I don't know any of these characters, I'm quoting you. And then it's a review, a perspective. She writes in I'm not going to get to the end of the talk, but it's okay, we can have a dialogue, that's good enough. Article, this appeared in 1984. And this were the opening several sentences are worth reading. So in case you get negative reports, Zedowska, in 1977, 7 years prior to this paper, Dan Dexter and I, with colleagues at Roger Williams General Hospital, I don't even know what that is, submitted a manuscript to Cancer Research. It's a journal that I published one paper in which we reported the isolation of 4 distinct tumor subpopulation. That's what I was showing you. Spontaneously arising in mouse mammary cancer. We speculated the subpopulation were evidence of intra-neoplastic heterogeneity. Nowadays it's simply called intra-tumor heterogeneity. And that the heterogeneity was a general phenomenon, knowledge of which was important in the treatment of cancer. Editorial review of this manuscript, if not swift, was unambiguous. It was worthy of publication. It was not unworthy of publication in Cancer Research. 2 reasons were given. Our results were not novel. And anyway, everyone knew that cancer are monoclonal, i.e. there's no intratumor heterogeneity. So they rejected this paper. She goes on to say,
aren't those 2 reasons contradictory? If everybody knew already the results were not novel, so because the results are contradictory.
Well, it turns out in the 50s, people knew about this problem, oncologists. They knew that this problem existed, but they didn't know much about this. So, as he was saying, the sources of heterogeneity are both genetic, it could also be epigenetic, post-translational modification of various things, and then also stochastic mechanisms. So these are all very nicely explained in the review article that I showed you, and Nelly Polyak has written other review articles more recently, a couple of years ago. It's an old idea. In fact, the people in glasses should be thinking about the cell guys that knew about this sort of.
But the one before last sentence is really fun. I could only reflect that I too was originally monoclonal.
Yeah.
But became undeniably heterogeneous.
Yeah. This article is extremely. Maybe I should only discuss this article. It's an extremely interesting article to read. And again, all these things I came across very accidentally. So really, the person who showed, even though it is known categorically clearly that ITH matters, intratumor heterogeneity matters, is an English physician, Charles Swanton and company. They published this paper just about 10 years ago in New England Journal of Medicine. And there they biopsied this person and other patients, 3 or 4 different cancers, I think. And they actually biopsied 7 regions. Those are R1 to R7. And the mutated genes in various regions are in yellow, and the ones that are not mutated are in blue. So this is like a matrix that we actually analyzed, but I'm not going to tell you about. And you see that the mutations in different regions can, in fact, be quite different. And hence, if you want to treat this tumor, you got to deal with it. And Mr. Swanton is not sitting back. He's done quite a lot of work, and it's got some implications for evolution of the disease, evolution of cancer itself. And in fact, he wrote an article that I don't have the reference to, which if you didn't know that Charles Swanton was an MD with whatever they call it in the UK, you would think that he's a physicist. It's really a beautiful language and precision with which he describes waveform. Okay. So, in the kinds of problem, like this colloid problem that I mentioned, Paul's stuff, we sort of know how to at least simulate the thing because interactions are reasonably well known. And then it's only a question of system size. You can bang on it with the computer, into Brownian dynamics, or do whatever you want because these are all motions over damped. And the question that we ask ourselves, knowing nothing in, let's say, 2014-15, is can we do something like that for cells? And if you knew too much, which is not our problem, then you would immediately say, look, single cell itself is a factory. Right? And in fact, you're working on some small aspects of that factory in terms of cytoskeletal rearrangements. It's crowded, lots of components and stuff like that. It's pushing and pulling on the membrane, which is communicated to the neighboring cells, etc., etc. And then, of course, the cells, like in the first movie I showed you, can divide, undergo apoptosis, which if you have cancer, the apoptosis rate is small, and then the cancer, the fitness advantage, because of these mutations that confers it additional fitness, takes over the cell, normal cells, and goes. It's a very complicated system. Okay. This group of cells, which in the epithelial normal tissues, is pushing against this extracellular matrix as is migrating. That's all a good thing. It's a normal course of development. It must take place. The extracellular matrix itself is highly complicated. It's highly rigid and hence very high viscosity if you want. And that can be modeled. In fact, there are sorts of models suitably modified.
It's collagen mostly.
It's collagen, but it can be some other things as well. But there are some proteins, fibronectins, and so forth. So the estimate of the viscosity, as it were, of this extracellular matrix is made, and depends upon the species, the cells. You know, it's after all biology, and a lot of things matter. And the viscosity, roughly speaking, was 1,000,000 times more than water. So it's pretty big. So we said we're going to ignore that. We're going to ignore the medium viscosity completely. So before I tell you the whole model, I want to tell you about a toy problem. This toy problem works like this. I create a cell, and if you want, in the physicsy language, you'll say it's a T equal to zero temperature. So it's not going anywhere. Right? It's going to stay there. It's all classical, by the way. Then you allow the cell to grow and then divide. Okay? When it divides, the daughter cells kind of come apart. There's an instantaneous, if you want, impulsive force for them. And then the two cells would then also grow and divide for this purpose of illustration. And I'm just assuming all the cells are equal sizes. So then it goes on like this. And what you'll see in this movie is, the movie is herky-jerky, it's not continuous, because the interesting things only happen when the cells actually divide, otherwise nothing happens. So let's look at this movie. So you can see that some cell division, which I prescribed, it's just growing like nuts. And at some point, it'll take over the whole cell. Now, this problem does not require any calculation or even a simulation. If I wanted to look at the mean square displacement of this cell, all identical, I have only one length scale in the problem, that's a size, and I have only one time scale in the problem, that's a cell division time, and therefore my mean square displacement must go diffusively with the diffusion constant that's given by the length scale squared divided by the time scale. Now, the whole process that is quote-unquote active system is essentially because I allowed for cells to divide, because the medium doesn't allow it to move. So cell division, therefore, is an important ingredient in the collective dynamics. That's what this shows. And this is just quantifying this. So I can skip that. So cell division is important. There are three scenarios. I mean, I think in my reading of the literature, for collective movement of cells, one of them is called the self-propelled Voronoi model, and I'll show you that in a bit. And the other is when the rate of cell division matches the rate of apoptosis. So it's one that's a homeostasis. And the third scenario, which is the one we developed mostly, is the imbalance between the cell division rate and the apoptosis rates. Since there's a lot of people here who don't know whites, I want to tell you this little story. So when I was still in Maryland, I invited Dave Weitz to come and give a talk. And I jokingly said he has half Boston working for him, but it was only about 9 years. And he talked about this paper. This guy, Tom Angelini, was his postdoc, and there was some other, and they were looking at the two-dimensional movement of this MDCK cells on a substrate, and they kept increasing the density of this by feeding it. There's a nutrient that allows us to. And they could measure things like the velocity of the movement as a function of the cell density, or volume area fraction, I should say. And you see it decreases and maybe approaches 0. And then they made some sort of a estimate of what are the correlated regions in this MDCK cell, and how is the length and how does it grow with cell density. And you can see it goes from about 10 cell diameters to 30 cell diameters as you expand this cell density. I should have written this in dimensionless units, but I didn't do it. And they also saw things like what the 2D Lennard-Jones fluid simulation showed, swirls, and so on and so forth. So then they suggested there's a growing correlation length, and that was something that was lower in the glass literature then, as it still may be the case. So little did I know about the importance of this paper. I thought we'd just been working on glasses, and I now finished working by 89. I thought, oh, this fits in with my picture of how this should be. It's a jam system. What else do you expect? Anyway, in retrospect, I'm here to tell you it's a very important paper. And the paper is very important only because it probably, I'm not 100% sure of this, at least in the biological context, started the role of active forces. It's also, it's not a paper. I don't know, they wrote many papers on this. This first scenario sort of captures the stuff that you saw, and it's from also a very influential paper from Christina Marcotti, Lisa Manning, Max B, who is now professor in Mount Houston, and they created what's called a confluent tissue in 2 dimensions. Confluent means the packing fraction is unity. There are holes and things like that. And they simulated that. It's molecular dynamics simulations, and and there are pentagons and hexagons and stuff. So occasionally we stare at them. They even found a rectangle, but these swirly type motions exist just like in 2D Lennard-Jones system. And they did say this is like glass, and in fact they quantified this. The arithmetic associated with the energy function is simple. Energy just minimizes the area to given a preferred value and minimizes perimeter, and there's a tension between the two. Write down essentially a overdamped equation of motion, mobility times the force that's generated. This force is non-additive. It cannot be decomposed in sum of pairs or 3 bodies and so on and so forth. And then they added this additional term, which is the activity term, self-propulsion term, which acts only on the orientation of the cells, vectors. And there's an additional self-propulsion velocity associated with it. The key thing they showed in a computer
simulation. The question is, N is what, diffusing? And it's just rotational.
Yeah, it is diffusing. It has a Brownian motion of its own. Maybe it's in the next slide. No, it's not. It is diffusing also.
Okay, and then I'm assuming these are, you take the collection of points, you take RIs, you do Voronoi, that gives you this areas that you plug in the
Yeah.
energy. And then,
So what they found is that this additional mobility term, and this is the important point, that the dynamics is determined purely by the geometry of the cells, in some sense, that if you take the ratio of the perimeter to the square root of area, and if you have no self-propulsion term, it undergoes what they call glass to fluid transition at some critical value of 3.8. There's a math problem here that may require some proof that this is correct. So now you can see that if you're in the fluid regime, these are the various trajectories of these guys. It's all over the place, and it's more local. It's here. Okay? So this transition can occur in this two-parameter space, but it's at zero mobility. It's jammed here. It's confluent, which means that the packing fraction is 1, there's no cell division. So this is more or less saying the same thing. It's found some use in some analysis of experiments, and you know, you look at those experiments and go, the theory fits, but at least it guides the experimenters narrowing what they may think is going on in the cancer cells. The second scenario, which appeared long before this, comes from the French group, as my good friend Johnny in 2010. They did basically a continuum theory and as well as this computer simulation. And this is what you get. This is like a 3D, if you want the tumor. So what is their model? And then there was a subsequent work again by Jean-Louis Barral and others. They found that if the cell birth rate and the cell division rate are equal to each other, certainly asymptotically long times, then the motion is purely diffusive. There is no glass, there's no solid. It just undergoes diffusion, and it's, it's just so. Ka is the cell division rate. Kb is apoptosis rate, and they equally get insulin space.
Is it not similar to your simple model in which you only had divisions and this generated diffusion?
It's different because in the toy model that I showed you, there is no apoptosis at all, and there's only division. But here there is, and temperature, there's some finite temperature. So it's different from what I showed you before. And the toy model I showed you was merely to emphasize that cell division alone can generate these active forces. In fact, we call this a self-generated active forces, and that can give you motion even in the absence of interaction or apoptosis. Yeah.
But I thought the fact that your toy model, as you call it, is confined is similar to having apoptosis.
No, it's not confined at all. It's growing without any boundary. Yeah. We repeated these simulations of Johnny, and this is our result, and we just get confirmed what they actually produced. When you have homeostasis, where the birth and death rates are the same, even the long time limit, leads to displacement is purely diffusive. Okay. So the scenario 3 that we created goes like this. We wanted to retain all the simplicities, but maybe say a little bit more. We know that the cells are membranes, but fluctuate in fact, or you can have solid membranes as well. And so it's characterized by some elasticity, and they've been measured in some cases. We also know that these epithelial cells, the good kind of cells, they stick to each other. So there are adhesive interactions, and they're mediated by proteins, mostly E-cadherin, and the cell is coated with that for some density depending upon what species you want to talk about. And then, of course, I already said that cell in fact can grow and divide. And the division time is not so relevant, but for some odd reason, because we didn't know much, we took it to be 15 hours, and that turns out to be not horrible. Eukaryotic cells are between 18 to 20 hours. And then there's apoptosis. And because we're interested in the model of tumor growth, we made the apoptosis rate a fraction, 1/20th, if you like, of the cell level time. So this is what this movie looks like. This is a 2D version, although what I'll show you is 3D. And so there's no boundary here as well. It's although you could do this in a periodic boundary condition, it's totally free boundary. And some of the trajectories of some of the cells are shown in black, and it's very clear they have very directed motion. Yeah.
Okay, I'm confused. So I didn't quite get the model. What's the elasticity doing here?
That's a repulsive interaction between the two guys. Adhesion actually sticks together.
Okay.
Yeah. So the elastic model we're using is
It's just like Hertzian repulsion.
Hertzian.
Okay, but and then sticky.
stickies.
And then what's the third ingredient? Sorry.
I'm going to tell you all the third ingredient. I didn't tell you.
I'm confused how you talk about MSD with cell division, because a cell disappears, becomes two cells. So what does it mean?
Yeah, so we're only calculating the MSD of the cells that have not divided at all. So if you start with 100 or 200 cells, some of them survive. So you start with enough numbers so you can get statistics, and those are the MSDs associated with this.
That's a biased sample.
Well, it's biased, but it's feeling all the microenvironment, and that's what happens.
But it's a measure of what, like, I mean, okay, we've defined a number, MSD. At some point, they're all going to divide, so it's going to disappear. So you can't talk about the T goes to infinity limit.
No, I mean, T goes to 2 to 4 to 5 solution times, not infinity. So if I simulate this for 10 days, and I actually do, that's because the cell division time is 15 hours, 3 months, say, right? What you say is probably correct. But in the 4, 5, 6 division times, there are enough guys that survive that we can run many initial trajectories, like 200, 300, to say a reasonable average. But at the end, I've told you that averaging is not a good thing anyhow. Yeah, so this is just to be the limits of what's happening. Yeah.
Oh, I have a naive question. When a cell divides, how do you choose which one to track?
Track both of them, but so when a cell divides, I thought you were going to ask me, what do you do with the daughter cells? And that's a good question. What we do is we isotropically arrange the daughter cells, but I can also arrange it in a directional fashion, and we have argued physically without doing any calculations. They would give the same reason for reasons I can tell you later. That's because of this total buildup of the stress associated with division. It relaxes on a time scale that's much faster than the cell division time, so it's not very interesting.
Also a naive question about the phase transition scenario 1 part. What happens at the 3.81 phase transition physically?
I mean, the nature of the phase transition you're talking about, they didn't analyze it, and since it's not my model, I didn't care. Okay. Right? But there may be some interesting things there. Oral transition, they need some critical exponents and things like that. But they're very young, and you should ask these questions. I'm very old, and I want to solve problems rather than get hung up on these sorts of questions. I no longer worry about whether I'm mathematically precise or but let me tell you, it's what you're asking is a good question.
But will he get a job? His studies.
My boss in New York would say no. It's him. All right. So I haven't fully defined my rules. I only told you about attractive, and I told you the cell division and so on and so forth. But let me show you some experiments. These are imaging experiments. But we found out after we submitted our paper to Professor Rex, and some nice referee said, "You guys are idiots. There are already some experiments supporting you. You should probably discuss this." So this was an experiment that was reported in this journal Oncotarget, 2015. They embedded this fibrosarcoma cancer cell in a collagen matrix, and they imaged this guy. And they have the same problem, by the way. Some cells divide, etc., as we do. But they do exactly what we do. They, in fact, track 150 cells. We track 200 cells. It's a phase contrast image. But notice that if I sit at the center of this, this is a 2D projection, although there are experiments in 3D, 3 dimensions.
How do you track the cells in 3D then?
Huh?
How do you track the cells in 3D?
They have a very difficult problem that they slice it and look at it 2 dimensions, just like my first movie. 3D tracking apparently is damn hard to actually do. And of course, that's an interesting computational problem. Given a slice, many slices like this, can you reconstruct the image in 3 dimensions? It's probably reality, I don't know. So if I look at this, the edges of the thing are a little bit ragged, but the size of this is about, I think somewhere it says, but 1.8 millimeters. I think somewhere later on it says, and this is on day 6. Okay? They start with normal cells, and then they introduce a little bit of this cancer cells. Among the cells that have survived, I think it's 150, if I don't remember, they computed this mean square displacement as a function of time on the 5th day. And that goes as t to the power 1.4. It's super diffusive. They don't have anything to say about anything shorter than whatever this time scale is. I think the cell division time is about 18 hours here, but I can't be sure about that. This imaging, it's a very beautiful experiment. This imaging also says that there are some cells which move very fast, fast cells, some move very slow. So everything that we kind of thought we see in glass-like systems is here, and except in glass-like system, which is not driven, your diffusion, or precisely viscosity, will decrease exponentially, and depending on who you are, it'll have an essential singularity with the Couchman density, the constant temperature. That's not so here. Okay? All right. And then this is another representation of the same data. So the 3 ingredients in our model, I'll put them all together here. The systematic forces, this is the Hertz potential and some for interaction. The only place biology comes in is cells can divide around the apoptosis. This is super naive, right? So there are some parameters here which we don't adjust. In fact, we can read off from some standard cell literature, and then there's one rate and another rate. There's a 3rd ingredient that is extremely important here, and that's a feedback. We don't allow a cell. How we came up with this 7 years ago, I don't know. We're just super lucky. We basically said things can grow exponentially all the time, and therefore there's got to be some cutoff, and that cutoff is provided by a mechanical feedback. We basically say a cell can only grow and divide. The pressure it feels is less than some threshold value. And that's a feedback that cuts off growth. I'll show you that. Okay, I already showed you this toy model of a fistula and some issues. We can talk later. Okay, that's a model. So, look, I'm supposed to finish in less than 10 minutes. So I'm going to skip all these details. And so these are the systematic forces, if you want, between 2 cells. The Hertz plus adhesion, and you combine all that to get a single minimum at some 8 micrometer, which is roughly of the order of the cell size. In here, the cell size is polydispersed, incidentally. It's not uniform, as was the case in the toy model. And these numbers are reasonably good in comparison with single cell force microscopy, in which forces are applied, and they figure out how much force is required to separate 2 cells, and these are roughly according to that. So the second is, how does a cell grow? And this is also very naive. Let's say we have a cell that starts with some radius. It grows like this. This is the rate of growth, if you want, and it's surface area, and you can do this in 2 dimensions as well. And once it reaches the mitotic radius, which is R sub m, and it'll divide, we say that the volume is preserved. That tells you what the diameter of the 2 daughter cells are. That's
Equal.
it's given by this.
The two other cells are equal.
Yeah, that's what we assume. Again, this is an unnecessary assumption, but then you have to decide if it's unequal, how much unequal. And so, in the spirit of the physics approach, yeah, I don't know from a court carrying physics. In any event, that's what the cell division is. But this pressure is important. Only the pressure associated with the ith cell is less than PC, the threshold value. Can this grow? If it's not, it stops growing, and that's only temporarily, because later on the cells can rearrange, and this pressure can in fact go down and grow again. So the fraction of time that a given cell grows is equal to the fraction of time it spends with the pressure less than the threshold pressure. So that's what this says. If the PC is, this ratio is infinite, that means the cells are always dormant, because all guys will have greater than PC, which is 0. That's no good. But in the opposite limit also it's no good, but then the cells will grow exponentially, and that's not what people observe in the cells to do, and anyway, it's not appropriate. The fact that there has to be this feedback, the controllers, although we are not aware of it, was already known about, I don't know, some many years ago, by a guy by the name of Rakesh Jain, who was in Harvard, and much more recently, Boris Schmidt. They knew about this. So the equation of motion is simple. It's just for damp motion plus biology, and the biology is that. So this is an example of 2 cells, some value of pressure PC, and you can see that the dotted line is PC, which is 1 times whatever number 10 to the minus 4, and only when the cell pressure is here, it can grow. Above that, it's down. Okay, so you can count this. There are 3 intrinsic time scales in the problem that I mentioned. One is the irrelevant time scale, which is this friction coefficient divided by the elastic modulus times radius. It's a deterministic time scale if you want, and that's very small. And then there's a growth time, which is 1 over tau, and then there's a apoptosis time, and this time scale satisfies this. And that's why it's not an unreasonable model for solid tumor. A hallmark of the growth of, and this is tons of literature on this, dating back to the 60s and so on and so forth, they've actually measured the growth, the number of cells as a function of time. It's initially exponential on time scale of the order of cell division time, and then it becomes algebraic. And that's because the feedback starts kicking in when you have cells. Because that's what this shows, and this is one of the comparisons. And so it goes from exponential to some power law, and the power law exponent depends upon all kinds of things, like it's completely non-universal in some sense. Depends upon the PC, the cell type, blah, 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 blah. But the fact that it's exponential for power law is I don't, in biology, I don't ever want to use the word universal. It's widely found, shall we say. Okay. So this is means for displacement, and then it goes back to this question on the surviving cells. What do I do? And what you find is that, and this is why it's like glass-like caging behavior that people know about. The means for displacement as a function of time. In the intermediate time scale, what does it mean? T less than about the cell division time. It's sub-diffusive, and then a much later time, it's super-diffusive. And in this measurement, it's what 1.3. So the number actually is not so relevant. And if you recall the experiment, which I think I'll show you again, the experiment is 1.4. And remember, we not only did not know that this experiment existed when we submitted the paper, we didn't try to adjust anything to get this. And theoretically, we've shown that at least within the confines of this model, the long time behavior of the means for displacement has to be super diffusive, and that's reported in Israel E or something like that. Okay, so this is I'm just reminding you that that we are doing not so bad, and if you're looking at only normal cells, is diffusion, and that's what Johnny and company told us 15 years ago. When you have equal number of cell division rates and diffusive, but this is not so here. Okay, this is again not so. Yeah. Let me.
So here the division rate is much higher than the death rate because it's cancer.
1/20 or something. But you can vary with that. And in fact, we produce a phase diagram in terms of this PC and this tau, and it's pretty complicated. The people who produced it in my group say they understand it, but I haven't come to use it. So it's
I saw they were speaking about homeostatic pressure, which is the state which is the pressure at which homeostasis is established, so division and cell death balance each
yeah,
other.
correct.
And you are talking about
The opposite limit.
the opposite, the limit when it grows.
Then it grows, and the division time is more than.
Your division overcomes.
Yeah. Correct. That's the difference.
But that's what cancer cells are supposed to do.
That's what they do in
Bad case.
Okay, so I've shown this already, and this is from the computer simulation and of this model that I told you about, and you see manifestations of heterogeneity, and there are Ted and I invented various ways, higher order correlation functions that we can calculate to quantify this, and we checked all those things, and so it's good here. So let me show you one more picture, and this is how the growth occurs in this model. All the cells are sort of blue. Okay, this is a movie that I want to show because it was made by this woman, Anne Bowen. She's dedicated to doing graphics in UT Austin. Not paid by anyone but the university. And what she generated, so let me show you the movie. This is the end of the tumor, and then we're looking at slices. The various colors represent different speeds, and as it moves, you'll see red, blue, white, and the interior is blue, dark blue. As you come to the periphery, it's hotter, and it's red, and again, this maybe was a surprise to people who are doing cell biophysics, but glass people sort of know this. And this is showing the same. I think I'm supposed to be done, right? So okay, so, huh?
Or all that.
So this is another point of heterogeneity. We're looking at the velocity autocorrelation function. In a fluid, the integral of that would be the diffusion coefficient in the linear response limit. As a function of some delay time, and these various colors are the various cells, and the red is when you're kind of close to the periphery, and we use some machine learning to figure out whether you mean the periphery, whether you mean the inside, but that's a technically irrelevant detail, I suppose. And you can see there's a tremendous number of variations. We could do the same thing with the imaging experiments that I showed you on the target, and that comparison works out pretty well. Yeah, it's a very similar. The last thing I want to tell you is the MDCK cell that I showed you a long time ago. That makes a living, and if you calculate the rotational velocity, if you look at this velocities, you can see vortices, anti-vortices in this picture, and you can quantify them. In particular, you can calculate the energy. Remember, in an inertial turbulence, like Commodore, for example, you feed in energy on long length scale. There's a cascade that's lower. It's much more complicated than that, but then it dissipates at a molecular scale, and then from this you can work out in the inertial regime what the scaling of the energy should be. And here, what you find, this is real experiment, by the way. It's not a computer simulation. But we did the analysis. We also did some similar kind of simulations. The Reynolds number is basically 0. 10 to the minus 6 or something like that. And if you calculate the energy spectrum, it's the integral of the velocity squared of k as a function of k in the middle region, this in 2 dimensions. What you find is decays as k to the minus 1.58. Very, very close to the 5/3 that's expected for 2-dimensional inertial turbulence predicted by Kretschmann, I don't know, ages ago, 60s. And then there's something else on a long time, and we could make comparisons with experiments as well. This is a very active field, quote unquote, of research under the rubric of active turbulence. And the appearance of this inertial turbulent like scaling, whether it occurs in the intermediate or long time limits, is, to the best of my knowledge, not understood. And why it's non-universal elsewhere, and what's the origin of that, does it depend upon the cell species you're talking about, and time scales and so on and so forth. It's really not quite well known. And we and others, like Johnny, Erwin Frey, lots of other people work on this problem. Actually, my good friend Srinivasan, who is here, he's not here today, in Austin, it turns out. He's being feted in Texas. So he told me he won't be here. He is a big expert on inertial turbulence. And I tried to get him interested in this. He said, forget it. So I'm not sure if he believes this or I don't know. Anyway, there's a lot of stuff to do in this.
But I just want to note that one thing is in 2D turbulence, you know, the Kolmogorov scale, you can get it to be, so you can get like 3 orders of magnitude on the X scale,
Yeah.
and the exponent is clear.
Over
like half a decade,
Yeah.
an exponent of 1.58, and then that's a very
You're going to have this limitation with the biological system.
exactly
always.
right, because
Yeah.
it's a very finite
Yeah,
small
you
sample
just-
with
yeah.
a small number.
Yeah. It's totally different, and therefore you could say, well, gee, if you had, but then you also have finite. We're not infinite systems, and so this is an issue. You've raised it a few times, and it's valid, but so you could just say it's just mere coincidence. And
Exactly, yeah. It could be a miracle.
it could be, but it would be difficult for you to prove that. In the meantime, there's this tantalizing result, which is interesting.
It would be difficult for you to prove that that number exists, because then there has to be a limit. If you want a number, 1.58.
Yeah, I know.
you have to
But
use the limit.
this is experimental analysis, and it's there. And these guys are reported in many other places where it's there as well. The fact that I have to worry about finite systems and made for corrections and things like that is out the window. I'm going to do that, and it's just not kosher to ask the question, I would say, in this context. In inertial turbulence, yes, right? So there's a bunch of conclusions. What I didn't tell you, which I talked to the graduate students about, is that the generic motion of glass legs, superdiffusion, things like that are around in many other places. We noted before in the active matter community, and maybe very generic of certain non-equilibrium systems. And that too is an interesting theoretical question. What classes of, again, it's a bit too mathematical for my taste anyway, what classes of equations of motion would generate things like this? That's a mad question. I'm uninterested in answering, shall we say, at my stage in life. With that, thank you, Shara. Thank you for listening.
Any questions in addition to what we asked before? Yep.
I have a question about the pressure. I mean, so you were saying that there are scenarios where the pressure of the surroundings changes the cell growth or division rate. I was just wondering if you thought about what, I mean, that's the idea, right? So basically,
No, it doesn't grow.
Yeah, it doesn't grow, but for an actual cell, it has to decide to stop producing stuff on the inside, so it doesn't continue growing, right? So I was wondering if you thought about any scenarios where the finite time scale of response to the external pressure would have some overlap in the time scale of these growth and division times, and your dynamical heterogeneity time. Like if there's a regime of your model where those kinetics actually would play a role, as opposed to instantaneously the pressure stops growth, something like that.
First of all, the pressure is changing continuously.
Yeah.
It can go below.
Yeah.
And at that point, it remains dormant for a bit, and then it grows again if the pressure decreases further. Right? What are you proposing, Louis?
So isn't there I would think that there's kind of like an induction time. So you're saying the
Oh,
pressure's fluctuating and that the cell should decide
Yeah, and
over some
I
memory, right?
there could be a delay time.
Yeah.
That I should take into account. And if you do that, we've done some work on that. What happens is you essentially postpone the dynamical behavior rather than completely inhibiting it. And in that, if you keep increasing some new things start showing up. And how do you control that is even in in vitro experiments not clear, and whether nature does that, I don't know. But there is one important point that I want to make. That's something that we just have been working on for a year. This model is super simple. It neglects the fact that the cancer cell is greedy, which means that it can only survive if the nutrients are around. So it actually grows blood vessels, right? And we should worry about the effects of nutrients and oxygen, things like that. And it can be done, and we are doing it. But the problem is that now you have more parameters and additional things to think about. But it's super important from the perspective. That's why people say that cancer cells love sugar. That's because it needs to survive, it needs nutrients.
Which actually was my own question. On the scale of tumors that you consider, there are no blood capillary.
There, yeah, I mean, I'm not sure.
Okay. I saw that 0.1 millimeter there are already some.
Yeah, there are, and it's not here actually, and so it must be included. And we're next year or two.
I have another question. For simpler cells, is that true that bacterial cells do not divide if you subject them to pressure?
This I don't know.
Because several years ago, I didn't follow this very carefully, but several years ago in the castle, we discussed that, and he apparently did experiments, and apparently they do not care about pressure within the reasonable.
I was going to say to you, bacteria is very tricky, and it's impossible to kill the bacteria. So you can subject it to lots of things, and then the spores are the same way, and they probably don't obey this.
Didn't Sak Junjun at UCSD, didn't he show in the microfluid devices?
That's exactly what Ida did, and apparently, it's not pressure controlled by something. I forgot the details.
Well, in confinement, definitely at some point they
Yeah.
stop, right?
Yeah.
But that bacteria is tricky. It's really very difficult to get rid of.
I guess that human cells are more tricky.
Not tricky, but they are more finicky. Bacteria will survive all kinds of stresses.
Because they have
Alright.
states of I'm dormant,
Okay.
we don't have
Wake me
that
up
state.
later.
All right. Send
Just
you
had a
the.
question.
Oh, sorry. Yeah.
You had, in some pictures you had showed us, we had a critical pressure that you said, before we knew about the experimental results, we chose feedback. In the pictures that you showed us, they were the results of that experiment, but you had cooked up the model before you had seen. Where did you choose that pressure from? Or did I miss anything?
We varied that. So what we knew was the exponential followed by power law. This is old literature. So we want to reproduce that. And we knew about this element of physical argument, I can't make it too small or too big. So therefore we must have something reasonable. And then we calculated on the fly what the pressure, typical values of pressure is for the parameters like adhesion, etc. So we got some, so we varied. Trial and error is what it is, experimentation.
Any other question?
I was just wondering, so in in tumors, in addition to higher pressure, they also have shown that actually they have higher temperature.
This I don't know actually.
Yeah.
I mean,
So that is important. So it was the idea was at one time that for detection you could actually use IR imaging.
I thought that the
Temperature, like actual temperature.
Yeah, like
actually,
actually,
I
yeah,
thought-
well, because they're super hyperactive.
Yeah.
The
okay.
So
metabolism
I
is
thought-
higher.
I thought that Mustafa said, for example, in Georgia Tech, has devised this heating as a way to kill the cancer cells. So I don't know what the temperature regime is, but we know that their movement depends upon local stiffness and things like that. But the temperature, I would be afraid to measure that even on a computer, actually, let alone an experiment. And besides, in the way we did this simulation, the temperature is more or less an irrelevant variable.
We know that even inflammation leads to elevated temperature.
Right, but kind of one of the biggest issues, right, for healing the tumors is the targeting of if you have a chemotherapy. And so people are trying, right, to figure out various tricks, basically how you make your drugs choose actually where they're supposed to go. So one thing that was like very much in by now, I don't know, 20, 30 years ago, was that they were trying to find how to access actually places of higher temperature, because for some tumors it's true that they actually have few degrees actually higher. So
We
it
don't
would be
know,
like,
right?
like
It is.
39 Celsius it would be inside. Instead of 37.
That's a big jump usually.
Yes.
Yeah.
Okay.
Plasma physicists obviously are interested in this approach.
No, they want to kill the whole thing, including the patient probably. I don't know.
All right, any other questions? Let's end Dave once again.